Okay, let's continue our discussion of aerobic and anaerobic respiration. So today I will not go into that much detail, but since it's kind of like a new and interesting topic, I'll just take today's class to understand, give you some notes on aerobic versus anaerobic respiration, the two types of anaerobic respiration, and the four types of like the different steps in the aerobic respiration. It will be kind of like an overview of like a recap of whatever we did, but assuming that it is going to help you to get this complicated things understanding these things once more. Before we start, also we looked into two types of the different types of organisms based on the carbon source and the energy source. What were the two types based on the carbon source? One which takes from the inorganic carbon and the other from organic carbon. In here, you can add that not just the inorganic, you can have any electron donor. So essentially the carbon source is the electron donor. So any other thing that can donate electrons will also come in here. For example, iron 2, right? Sulfide, reduced ammonia, nitrite, right? Anything that has the potential to donate electrons will come in here. So it could be reduced iron 2, it could be sulfide, things like that. In here, it's very simple, the organic carbon. So this is atypically the sugar, right? And what is the inorganic carbon? Atypically, the carbon dioxide, correct? So depending on the carbon source, you have two types. What do we call these two things? Right, so these will become, what will be this? Speak up. And this? Based on energy source, what are the two primary? So light and redox. The chemicals, the redox. This will be termed as so till our discussion till now, we have been looking at what? Also, when we were looking at respiration, we typically look for the electron donors and the electron acceptors. So this, the respiration, could be based on the electron donors, which we saw there, right? So in here, we could have different electron donors and different electron acceptors. What is the most common electron acceptor? Most common electron acceptor, O2. This is the most common electron acceptor. And we will call this kind of respiration as aerobic. Respiration. In here, so this is one. We could also have anaerobic respiration, correct? In which you don't do, the electron acceptor becomes something other than the oxygen. So this becomes anaerobic respiration. Examples could be, what can accept your electrons? Sulfate can accept electrons. What else? Water is already reduced. So oxygen is accepting the electron, and we'll see that, and getting reduced to oxygen. So water is a byproduct. Water itself cannot accept the electron. Sulfate can do it, perchlorates can do it, nitrates can do it, right? The electron donors we already saw. The electron donors could be inorganic and organic. But the electron acceptors could be these two. Now, we will start with this, the aerobic respiration process. This is what we have been talking about, and how this has the 4 different steps: the glycolysis, the production of pyruvate, the TCA or the Krebs cycle, and 4th is the electron transport chain, right? So with that, let's start our discussion with what we already know. Just a recap of, so what are we talking about? We are talking about aerobic respiration. That's what we are talking about. So we are talking about aerobic respiration. What would be the chemical equation representing aerobic respiration? What do you have? Plus O2 will produce CO2 plus water plus energy, right? I'm not balancing it out. So this is aerobic respiration. This is what we are going to actually study. And we are trying to see where things happen. So we already know this. What do we know? It has 4 steps. First is glycolysis. The second is production of the pyruvate. What is the third? And what is the fourth? The electron transport chain. And there is a process that happens as chemiosmosis. It's very simple, but we'll see that. What happens in where does glycolysis happen? Right? It happens in the cytoplasm. What happens in glycolysis? So you have glucose molecule. This is a C6, correct? You produce how many pyruvates? Do you produce pyruvate or do you produce the acetyl-CoA. So in here I made a mistake, right? So this is acetyl-CoA production from the pyruvate, right? So glucose is a C6, it produces pyruvate. This is a C3. There are 2 pyruvates produced in this. What is the net ATP gain in this reaction? So just to write down, remove this and start writing. So net gain of 2 ATPs. Also, I want you guys to know this, that there are kind of batteries present in our bodies. You will hear about 3 types of batteries. One is the ATP, the other is NADH, the third is FADH2. These are the 3 batteries. Now these are the charged batteries. The discharged batteries of these corresponding batteries are what? What is the discharged version of the ATP called? ADP. What do you need to produce this? A phosphate group, right? NADH, what is the discharged version called? It's NAD plus. And this, FADH. These are different types of batteries. We will see this in fermentation, in which we do not have a lot of we don't have oxygen. We will see that these discharged batteries play a very important role, because we do not have a lot of discharged batteries in our systems. The availability of the discharged batteries will determine how many charged batteries you produce. It becomes a little bit of a different problem. It does not depend on the amount of glucose that is present. So you might have a lot of glucose present, but if you don't have oxygen, you will not be able to produce a lot of energy, and we'll see that. So it will not matter how much glucose you have. If you don't have oxygen, this entire process can sustain up to some level and then it stops. We'll see that. Now, we also get some amount of other batteries. What is the other batteries that we get in glycolysis? So you get 2 NADH. And how did you get that? You reduced NAD plus into NADH. In that entire process, you reduced NAD plus into NADH. How do you get reduced? So what is happening to the NAD plus? It is gaining electrons or losing electrons? Gaining electrons. So this is electron rich. Remember that. After all, everything is electrons, right? This is where some of the electrons are going and saving its energy, right? So NADH. Okay, and we will see how this NADH or FADH2 is going to play a role in then producing ATP. This is the most important one. We'll see how these and these finally help in producing these ATPs. So this is some of the other smaller batteries that are actually helping you to produce the ATP. Okay, we'll see that. Also, this is the process that is common Glycolysis is the process that is common in both aerobic and anaerobic respiration. So this process will also happen common for aerobic and anaerobic aerobic respiration. So whatever you do, you are actually producing two pyruvates. So this is the product of glycolysis. Okay, this is the first. Now, the second process is using this pyruvate and producing using the pyruvate and it is associating itself with an enzyme and producing this very important compound called the acetyl-CoA. Okay? Now, we saw that initially in our last class that fatty acids can directly produce the acetyl-CoA. You can directly break down a protein so you can have the protein and you can directly from the acetyl-CoA without forming the pyruvate. There could also be proteins, so fatty acids and proteins can both produce acetyl-CoA directly, but some of these proteins can also produce the pyruvate and which can then produce these. Okay? So you can have fatty acids that can directly produce the acetyl-CoA. You can have proteins that can actually go to pyruvate and then do this, or it can directly come here. So all these are food sources, but you will see how the fatty acids or how the fats differ from the sugar in this manner. They don't do glycolysis. Fatty acids are not getting involved in glycolysis. Okay? I'm not getting into the details of that. After this, let's look into in the acetyl-CoA reaction, how many ATPs are getting produced? And where is this happening? So the glycolysis was happening in the cytoplasm. Where is this happening? When it is entering the mitochondria, not exactly. So while entering the mitochondria, this is where you have this. This thing, so pyruvate starts to lose. So this was a 3-carbon, and in here you remove a carbon as carbon dioxide. The remaining 2 carbon containing compounds associate with the enzyme, and then you get the acetyl-CoA. Okay? Now, the energy output is you don't produce any ATP, but you produce 2 NADH. Okay? Now, moving into the Krebs cycle. Now we are in the mitochondria. When you are in the mitochondria, you now have access to the acetyl-CoA. And this is the mitochondrial matrix. is happening inside the mitochondrial matrix, and the acetyl-CoA is completely burnt. And this is a cycle, okay? So I'm not describing the entire cycle. This goes through different kinds of carboxylic acids. This acetyl-CoA gets converted into carbon dioxide very simplistically. This is what is happening. So this is the complete breakdown. This is why you are breathing out carbon dioxide. Okay? What is the energy output of this? You get 2 ATPs, 6 NADH, and 2 FADH2. So in here you can see that all that sugar that you started off has already produced the carbon dioxides that you are breathing out. But the amount of ATP that you have produced till now is a meager 4 ATPs. 2 in the glycolysis step, 2 in here. You have some of these batteries produced, but the ATP, which is the main energy molecule, you don't produce a lot of that. ATP is still here. The next step, the electron transport chain, is the main location of ATP production. Okay? But in here you will see it is producing a lot of these charged batteries. And charged batteries are what essentially? What are these? These are electron carriers. So the main agenda of the Krebs cycle is to produce these electron carriers, not the ATP. The ATP is a small 2 ATP, similar to glycolysis. The main agenda here is to get these electron carriers charged up. Because these electron carriers will now dump their electrons here and come back and get charged again. Dump here, get charged up again here. Dump here, get recharged. So that's what is happening. So these electron carriers will not only get, there are a limited number of these electron carriers, right? So like the number of discharged batteries are very, very small. So they charge the batteries here, discharge it here. Then again charge it here, discharge. That's how it goes. The main action happens in the electron transport chain. Okay? Now, let's try to understand what's happening in the electron transport chain. Just remove this. So this is happening inside the mitochondria, right? So imagine that you have all these different, what are these? What do you think these are? What am I drawing? So what could these represent? Yes, so this is the organelle membrane, whatever, right? Then you will have these complexes. We saw these kinds of things in the electron transport chain, right? Gate 1, gate 2, gate 3, and gate 4. And in between, so this is the fatty acid that is making the cell membrane. There are different names to this. I'll just write it here. So this is called the ELTH, the complex 1, complex 2, and complex 3. Easy to remember. What these are going to do are to discharge these electron carriers. That's their main job of these complexes. Now, there will also be so you get an NADH, a charged battery coming close to this. What happens is it dumps its electrons to the first gate. Okay, so you have this electron and it dumps its electron to the first gate. When it dumps these electrons, it is actually creating an electron gradient, right? So it is dumping its electrons. What happens then is this electron then gets transferred to the next. This electron is then transferred to this, then it gets transferred to this, and then it gets transferred to this. So the same electron is now moving from one gate to the other and sequentially it is losing its energy. So it's a highly energetic electron and then sequentially it is losing its energy. Okay? Now, when you get to these low energy electrons in here, so this is a low energy electron, someone must be present to handle these low energy electrons, because lose electrons in the body is not good. Who do you think is going to accept these electrons? Oxygen. Oxygen is the electron acceptor. So what will oxygen do? Oxygen will take the electron and it will take some of the H plus that is present here and gets converted into what? It will produce water. You can balance this. In the meantime, you also get other, so FADH2 is also donating its electrons in here, in this entire process. So there's a huge dump of electrons that is happening on these areas, right? Now, because of this, this you can think of as a cascading bucket of electrons. So you have a huge bucket of electrons in the top and you dump the electrons into the lower bucket. And then that bucket dumps its electrons to the lower bucket, and so on and so forth. But this electron has to be gone by the time you get to these complex 3. Okay? Now, the protons are going to be really important in here, because these are the electrons and protons are what? Just the opposite of these electrons, right? So if you are getting all these electrons in here, you will start getting a lot of the protons on the outside. So there is a proton gradient that is being established on the outside. Also, you will see is a lot of the proton is getting consumed in here. So what do you think is happening? Is there a proton gradient? Like if there is a proton gradient, where do you have a lot of the protons? Inside of the cell or outside of the cell? So this is the outside of the cell. So this is outside.